All right. Hello, everybody. Welcome to the next section, plant-water relations. Don't forget, in plant-water relations, we are going to discuss four main sections. We're going to discuss the properties of water. We're going to discuss SPAC, soil plant atmospheric continuum, and then in that plant section we're really going to pull out and understand the xylem and how it functions and what also could potentially hinder the xylem. Very interesting. Now, the first thing is I have given you YouTube videos, and that's just, I will explain them as well, but it's also just to help you guys grasp the concepts better. This section is quite a difficult section to grasp. So this first video on water potential is a very enthusiastic guy, and he just talks you through it. He talks you through what water potential is. So pretty much everything that I am now going to discuss is summarized in this YouTube video. So I suggest you pause this, you go watch the YouTube video, and then once you've watched the YouTube video, come back, and then I think it'll much easily explain. So watch the YouTube video, it's 10 minutes. Come do these slides and then go back to the YouTube video. It'll really put things in a very nice bow. But before we get on to plant water relations, we really have to understand the unique properties of water, because if we don't understand the unique properties of water, we're not going to understand what makes water so unique. So the first thing we need to remember is the polarity of water. Now we're going back to Chem 110. If you are anything like me, I just passed Chem 110. So I hate chemistry, and my major field right now is biochemistry. So the irony of it all. But anyway, the polarity of water. So when I say the polarity of water, remember we have water is made of H2O. We have that strong oxygen that has a very negative potential, and we have those hydrogen being positive charges. And because of the positive and the negative, remember opposites, they push away from each other. They become negative and positive polars. So we've got these two regions over here. We've got a region, a very negative region and a very positive region. So the polarity of water, think poles. Polarity, we've got a north pole and a south pole on Earth. We have a negative pole and we have a positive pole in water, and it's the structure of this water that's very, very unique. Now, there are also two types of bonds. Now, please, you cannot get confused. There are two types of bonds that are very important. Remember from Chem 110, bonds are the connecting of molecules or elements together. So the bonds that connect the hydrogen and the oxygen are called covalent bonds. So the bonds between the hydrogen and the oxygen are covalent bonds. And remember, covalent means they're sharing. So they're sharing the electrons, but oxygen is a little bit stronger. So hydrogen has one electron that it can give, and oxygen's a little bit of a bully, and that electron tends to stick with the oxygen more than the hydrogen, and that's how we get this negative polar and this positive polar. Covalent bonds sharing the electrons. Oxygen's a big bully. It shares this, most of the electron stays on the oxygen more than the hydrogen. And the second type of bond that is important for water is a hydrogen bond. So the hydrogen bond is between two molecules. The hydrogen, the positive hydrogen from this water molecule will form a hydrogen bond with the negative oxygen on this molecule. So you've got covalent bonds within the molecule, and those covalent bonds are very, very strong. And this is going to be very important later in photosynthesis. So remember, you look at these sections, you understand each section in its own box, but you need to have the capability to interlink and combine and connect each lecture slides with each other. So this hydrogen bond between the two molecules is a lot weaker than the covalent bonds within the molecule. Water is also a solvent. It is the universal solvent, which means things can dissolve in the water. The last thing is this idea or concept of latent heat. So as the bonds break, or as energy is put in to break the hydrogen bonds or the covalent bonds, whatever the case may be, most of the time it is just the hydrogen bonds, because those covalent bonds are super strong. It releases energy. And it's in that release of energy that transpiration becomes very important. So in the plant, by breaking those hydrogen bonds between the water molecules to change it from a liquid to a gas form, it takes and it expels the energy. So this latent heat puts energy into breaking it, and that extra energy, it comes from the sun, from the photons hitting the leaves. So when it's too hot, puts the extra energy into breaking these hydrogen bonds and sending the energy out through the stomata in the form of gaseous molecules. That's what we mean by latent heat, and that's important for transpiration. So that's how transpiration cools the plant down, by breaking those hydrogen bonds, which takes energy. So using that excess energy, and then as a result, it results in cooling the plant down. Okay, so those are the main properties of water that's important. Now, when you were younger, please tell me that none of you did it. I did not. I most certainly did not. But you've got those horrible people that and if you were one of those people, I do apologize, but that's slightly sadistic. So if you've ever put salt on a snail or a slug, what happens is the snail or the slug dies, and it dies because it draws all the water out. So remember, now we'll get onto water potential, but here we go. You can see here's our water molecules. Here's the water molecules over here. A bunch of Mickey Mouses are my water molecules. They are forming a nice region. This is my snail. So this is inside my snail, this box, and this is outside my snail, and this is the snail skin over here, the snail skin. So remember, water is a solvent, and things dissolve in it. So here's my NaCl, here's my salt. I'm going to put my NaCl onto my snail. So it's going to go into this box over here. And remember, let me just show you. Remember, the water being a solvent, it's going to dissolve, so that the Cl and the Na plus are going to come apart. And you're going to see here, the negative of the oxygens are going to be attracted towards the positive. Opposites attract. The positive hydrogens are going to come to this chlorine of the salt, and you're going to start seeing here we have gaps. We have gaps over here, and because we have gaps over here, the water is going to flow from the snail skin, or from the snail through the skin and out to the salt. So you've actually desiccated the snail. So just to reiterate everything in the slide, because it is very important to understand, we have put a solute, the salt, onto the snail skin, and the solute has made a negative or more negative water potential, and water flows from a high water concentration to a low water concentration, from a less negative water potential to a more negative water potential. So all of a sudden you can see now the salinity creates the Na and the Cl creates these gaps over here. So all of a sudden we've got a low concentration, we've got a high concentration. So the water will flow from the snail out the snail to fill these gaps over here, because the salinity makes it more negative. Now it is a lot more complicated than that, and we are going to get into it, but that is just the basics of it so far. The unique properties of water, how it will interact with the solute, and what being mean to a snail and trying to kill a snail effectively, what you would do. Please, guys, don't go doing experiments on trying to kill snails. I know you're bored in lockdown. Listen to the lecture. Okay. So this guy over here is King Poseidon, depending whether you're Greek mythology, Latin, whatever the case may be, Roman, whatever. This is Poseidon. And Poseidon is king of the sea. And when you think, well, you see now, Poseidon, I think of the Triton, but some of you from Ariel, her dad, that's fine too. Disney, if you wanna do that, that's your just, then that's what you do. But Poseidon really has Poseidon. It's psi. So this symbol over here is the Greek symbol for psi, however you want to say it. Just don't call it the little forky thingy. But yes, so this is the equation. Now, highlight this slide. I don't know. You're gonna have to you have to know this equation. There's no ands, outs, buts about it. You have to know this equation. This is a very important equation for the rest of the section. And if you plan on doing plant physiology next year, or in the future, this equation is the water potential equation. And before we get onto the water potential equation, it's the potential energy of water per unit area compared to pure water. Just the potential in an area. So remember, if I bounce back here, the NaCl created more spaces. It used less area. So we went from an area where we could have more going in. So this is the equation you need to remember. Water potential equals solute potential plus pressure potential plus gravity potential. Now, gravity potential is very much in red here. When you are looking at a cell, the water potential of a cell, gravity is negated. There is no gravity on the cell. So I'm going to say that again because this is very important. When you're looking at cellular level of water potential, when we're starting off at the roots, and we're looking how the water moves from one cell to another cell to another cell, gravity potential is negated. It's left out. It's ignored because there is no gravity affecting at a cellular level. Having said that, gravity will definitely play a role in a giant tree. So this is the stratosphere giant. It is the tallest known living tree, and that is quite high. When you start getting to this point and you're looking at the water potential in the xylem, for example, you have to take, especially at the top levels, you have to take into consideration gravity. But for this module, when you are looking at this equation specifically, we are talking about it at a cellular level. And because we are talking about it at a cellular level, you can negate that gravity potential. So ultimately, your equation is going to look like this. Water potential equals solute potential plus pressure potential. Now, I get very, very confused in this section. And I get very confused because of the high water potential to a low water potential, from a high water concentration to a low water concentration. It gets too much. So and now, because it's going to be online, I can't see you, but perhaps you want to have this slide pasted somewhere on your wall, so as you're going through the lecture slides, as we're going through it, you can continue coming back to the slide, because water moves from less negative to a more negative water potential, from a high water potential to a low water potential, from a high water concentration to a low water concentration. And you need to remember that water potential is always going to be negative unless it is osmosis, or unless it's homeostasis, where it's equilibrium. The solute potential is always going to be negative. So because remember, the solutes make spaces, and because they make spaces and they hug the water molecules, it makes the water potential more negative to allow for more water to flow in from a higher concentration to a lower concentration. Pressure potential is always positive when you're looking at the cellular levels when it comes to water potential. So if you're adding pressure, it becomes positive. If you're taking pressure away, where it becomes flaccid, you may go into negative zones. But ultimately, pressure, when you're adding pressure, it is positive. When you're adding solutes, it's negative. And when you add solutes and you keep the pressure constant, the water potential becomes more negative. If you keep your solutes constant and you increase the pressure, the water potential decreases. So you might even want to do arrows here. So as the solutes increase, the water potential increases. As the pressure potential increases, the water potential decreases. This is so important. This is going to set you up for the rest of the section. You need to understand this. Water potential moves from a high water concentration to a low water concentration. Solutes, an increase in solutes will increase the water potential. And this is why I get confused, because you would think that if it's increasing the water potential, it's increasing the water potential. It's not. It's going to make this water potential more negative. And that's how I'm going to refer to it throughout this module. I'm going to refer from moving from a less negative to a more negative. So as the solutes increase, it becomes more negative, and in becoming more negative, it makes the water potential more negative. As the pressure potential increases, it becomes positive, it makes the water potential more positive. So if we have a look at this equation, we're simply going solute potential plus pressure potential. They're both zero at this stage because it's pure water. There's no solutes in it. There's no pressure. There's nothing being put on the top of the beaker to make it pressure. It's exposed to atmospheric pressure. So it's going to be 0 megapascals. Having a look at a solution containing 0.1 molars of sucrose, I will show you how you calculate that negative 0.244, but it's calculated to give you negative 0.244 megapascals. Still no pressure is being added, and because we only have that, remember the solute potential is always negative, so that water potential is going to be negative 0.244 megapascals. Now, how did I calculate that solute potential value? Well, again, for those mechanical engineers who think they're the bee's knees, we have this concept of the ideal gas law. And you should have been introduced to the ideal gas law in chemistry. Do I expect you to know the ideal gas law off by heart? No, absolutely 100% not. What I do expect you to know about from this slide is that the ideal gas law gave rise to Van 't Hoff's equation. So I'm going to say that again. I don't expect you to know the ideal gas law. I expect you to know that the ideal gas law gave rise to Van 't Hoff's equation. Yes, you do have to know Van 't Hoff's equation. No, I am not going to tell you, here are the values for Van 't Hoff's equation, calculate the solute potential. I am going to say, explain to me Van 't Hoff's equation. And you're going to have to explain it to me. I'm not going to give you the values and you need to calculate. That's way too easy. That might come up in a prac somewhere. It might not, but it's definitely not going to come up in the exam. I'm rather going to say, what is Van 't Hoff's equation? And then you're going to have to tell me, well, it's derived from the ideal gas law. This is the equation. Solute potential equals minus RT CS. And these are what each letter means. So for R, the gas constant, there's a gas constant here in the ideal gas law as well. Sorry. And this is it. The temperature is measured in Kelvin. And remember, this is the equation to get the Kelvin. There's Kelvin and temp and degrees are different. The Americans use the Kelvin system. Shame. Anyway, then that CS, oh, okay, see here, let me fix that right now. This happens every year. Okay, that's C. Needs to be a big letter C. What do you mean no? There we go. Okay. I will click save. Sorry for letting me do this. It does it every year, changes that. So this SC is osmolality, or I or M. So remember, we said up here, we had 0.1 m's of sucrose. So osmolality is how much of the moles is dissolved in of the solute per liter of water, or in the solvent, moles per liter. Now, the reason why I'm not going to give you equations to calculate, it becomes a lot more difficult when you're dealing with ions. So, or ionic solutions. So remember, for sucrose, there's not really any ions to deal with. So you can comfortably say 0.1 molars. But when you start looking at something like salinity, which has a positive anion, and a negative ion in the chlorine, you've now got to account for the fact that that's going to be times 2. So whatever that 0.1 is, times 2, because it's separating into 2 individual ions. So that is why, the main reason why I'm never going to ask you to calculate the solute potential, because that osmolality becomes really difficult. So what do I expect you to know then, if it's a bit too difficult for second year? What I expect you to know is the solute concentration, or the solute potential, is measured by the Van 't Hoff's equation. The Van 't Hoff's equation was derived from an ideal gas law. We have a constant, which is the gas constant, represented by R. We have the temperature, which is measured in kelvins, and we have osmolality, which is the moles of total dissolved solute per liter of water, and ions will make it times by 2, because, or depending on how many ions are released into the solvent. So, and remember, we measure Van 't Hoff's equation in megapascals. You will see other in the extra readings or in lecture slides, not lecture slides, in YouTube videos, you might see something other than megapascals. I want you to remember that this will be measured in megapascals. So, solute potential, always negative, and it's negative here. Now you can see because of that R of this equation, where and how it is derived from the ideal gas law. All right, I hope that makes sense. Do not panic about being able to calculate anything. You do not have to calculate it, you just have to understand what the equation calculates. All right. So we started looking at water potential and how we calculate solute potential. Let's just have a look for a quick minute about pressure potential. Now, remember, pressure potential is going to, if there's pressure being added, it's going to be positive. So here we have a beautiful plant cell. You can see the beautiful chloroplast, mitochondria. It's a very pretty cell, a plant cell. What enables the plant cell to be able to withstand pressure is the cell wall. The cell wall is lined with lignin, which is a polymer complex which enforces or reinforces the cell wall. And by reinforcing the cell wall, makes that structure of the cell wall super strong to allow for the plant cell to withstand the internal hydrostatic pressures, whether they be positive or negative. Remember, when a plant cell has water coming in and it becomes nice and tight, it's turgid. When there's water leaving the cell, it becomes flaccid. And when you've got a process of water coming in and leaving at the same time, you're really in a state of homeostasis, isotonic. So pressure potential really helps with cell enlargement, gaseous exchange in the cell, because remember, we have to get carbon dioxide to the chloroplasts to be able to do photosynthesis. Pressure potential also allows for transport and also to be rigid, to really ensure that the cell remains strong. And all of this cannot happen without the cell wall. If the cell wall is not there, the cell will collapse in on itself. So if there's too much pressure coming into the cell, it will burst. If there's too much pressure leaving the cell, it will collapse on itself. So remember, the cell wall, cell enlargement, gaseous exchange, transport, and being rigid. And it really allows, and it's a cell wall that allows the pressure potential to be able to be withstood by the cell, the plant cell. Okay, pressure potential will generally be measured by something called a pressure bomb, which you will get to, which is in your pracs as well, in your prac manual. Okay, so let's just clarify some terminology quickly. Some terminology of diffusion and osmosis. So diffusion is spontaneous movements from a region of higher to lower concentration, and diffusion is a dominant mode of transport. Now, another name for diffusion is osmosis. And osmosis is diffusion of water across a selective permeable membrane, those cell walls or the membrane covering, or whatever organelle is covering that specific, whatever membrane is covering that specific organelle. But generally, when we say permeable membrane, we're referring to the cell wall. So diffusion, the movements of water specifically for plants at cellular level, osmosis is another name of diffusion, but now we have a selective permeable membrane. So if you say diffusion from one cell to another, that is correct. The more correct answer would be osmosis. And osmosis is just a different type of diffusion where there is a selective permeable membrane. So we looking as osmosis, and when you start looking from cell to cell movement, we really refer to osmosis. So when the water enters through the root, it's going to move via cell to cell to cell to cell until it gets to the xylem. And once it gets to the xylem, we're going to deal with bulk flow. But for the time being, osmosis is from cell to cell to cell. Now remember, there are two main ways water moves through a plant. The first one is osmosis at a cellular level, where we go from cell to cell to cell, and the second is bulk flow, and that generally happens in the xylem, where we go from the bottom to the top. So, two ways, osmosis, bulk flow. Osmosis is just the diffusion or the movement from a high water concentration to a low water concentration, or from a high concentration to a low concentration through a selective permeable membrane. Selective permeable membrane generally being the cell wall. So here is a very nice equation of water potential equals solute potential plus pressure potential. Now remember, as water around here moves into the cell, you're going to have more water coming in, and this pressure potential on the cell walls is going to be exerted. There's going to be a pressure exerted because water is coming in. So you have a solute in the cell. The solute's -2, and it's drawing water in. And then all of a sudden, you're going to reach a point where the pressure potential equals the solute potential, and you have an equilibrium. You have a homeostasis vibe where the water's coming in and it's flowing out, and it's keeping the plant nice and turgid, nice and strong. So yes, as water flows in, your pressure potential is gonna increase, making the water potential or negating or cancelling out the effect of the solute potential. And then let's say, for example, watch my mouse. I'm going to the bottom of the screen here. There's a cell down here, and it's reached a stage of 0. It's reached a stage of 0, and this cell over here is still -2 plus 0, so the water potential is -2. So the water is going to move from this water potential of 0 to -2, so the water is going to move this way. And as the water moves this way, it's crossing through a cell wall. It's passing through a cell wall, and as it's passing through a cell wall, it's coming into the cell. And this process is known, yes, you can call it diffusion, but because there's a selective permeable membrane, the correct answer is osmosis. It moves via osmosis into this plant cell, making this plant cell gain a pressure to where it's going to equal this negative solute potential until it reaches 0. Now all of a sudden, this cell down here at the bottom has got a negative potential again, and it's going to take from the cell at the bottom of it or on the side of it, it's going to take water back in that way. And then up at the top here, we've got another cell that's now -2 because it's got a solute, and it's going to take water from there, and eventually it's going to go from cell to cell to cell until we get to the xylem or the Caspadian strip, which we will get to. Okay, so to recap this ultimate section. We had a look at a cellular level, and at a cellular level, we considered the water potential equation. And because we considered the water potential equation, we had to look at each aspect. We know how to calculate solute potential. We understand how pressure potential works. We understand how it influences the water potential of a cell. We're not forgetting that this first little section was how water moves from one cell to another cell to another cell via osmosis. We also had a look at the properties of water. Now we're going to start moving to the plant as a whole. And this slide is a brilliant slide because it sums up your whole section in one beautiful picture, and it is stunning. Now, in the introductory video, I said an acronym, SPAC, Soil, Plant, Atmospheric Continuum. And this is a beautiful picture that shows you how water gets taken up from the soil into the root hairs, passed via cell to cell to cell towards the Cascadianship. It's pumped into the xylem, and the xylem will take the water up to the top of the plant, to the leaves, and again by osmosis till it gets to the stomata, and latent heat is released and transpiration out through the top there. This, if you can take the different aspects of this beautiful diagram here and explain everything on this diagram, you will pass this section. It's a little bit more complicated than that, but perhaps you want to print this out, draw it, whatever, and along with your water potential equation with the from a high to a low, whatever, you're going to stick it up there too, because you'll pass this module if you can explain what's happening in these equations or in this picture. So beautiful picture. Remember, if you have a look here, you'll see the soil water potential is sitting at negative 0.3. This root xylem is sitting at negative 0.6. And as you go up, and until you eventually reach the outside, which is minus 100. So you've got this idea of water wanting to move from a state of less negative to more negative, from a high water concentration to a low water concentration. And because of this movement, we're going to get movement or water moving from the bottom to the top. Now, a lot of plant physiologists will generally either relate to one of these next following. They'll say this is more, this is more. I am of the opinion, please bear in mind, this is my opinion. The research is still out there, it's still undergoing, but there are three main theories on how the water moves from the bottom of the plant to the top of the plant. The first is this idea of transpirational pull. And you can clearly see the transpirational pull because the water's moving from a less negative to a more negative. So as the water is leaving, the transpiration is letting it out. It wants to pull more water up, because remember, as it releases water up here at the top, it's going to become more negative, pulling the water from the bottom to the top. There's a second theory, and the second theory is really about positive root pressure. So what happens is you've got the water down here at the bottom coming into the root cells, and as they come into the root cells, they become turgid, nice and strong, and they create a positive root pressure, and this positive root pressure essentially pushes the water up the xylem. And then the third theory, which is the more popular, I believe, is the cohesion-adhesion theory. And this cohesion adhesion theory really arrives and derives itself from the properties of water, being able to adhere to cell walls or cohesion to each other. And because of this cohesion and the transpirational pull and the root pressure, it moves up like that. So, a lot of plant physiologists are also of my opinion that it's a combination of these three theories. Some believe only in one, some believe in the two, some believe in that one and not that one. I am of the opinion that it takes all three theories to get the water from the bottom to the top. You need to know this. You need to be able to tell me how the water moves from the bottom to the top. You need to be able to say there are three theories: positive root pressure, cohesion-tension theory, and transpirational pull theory. You need to be able to mention those three off the bat. I do not need to hear my opinion, so you do not have to say to me, because it's Candice's opinion, I must put all three. They are working theories. They are all three working theories: the positive root pressure theory, the adhesion cohesion theory, and the transpirational pull theory. And I believe it takes all three, or a combination of all three, to get from the bottom to the top. So now when we start looking at SPAC, soil plant atmospheric continuum, we're going to start at the bottom and take it to the top. I think there's a song. I started at the bottom, now I'm here. Yeah, whatever. Okay, so going to start at the bottom with how the water goes from the soil into the root hair to the xylem, from the xylem to the top, to the stomata, and out. And we're going to learn how this process happens and how this works. Please do not forget that between the roots, the water coming into the roots to the xylem, and from the roots, from the midrib or the veins of the leaves to the stomata, we have osmosis, water flowing or water moving via osmosis. From the bottom to the top, we have it moving via bulk flow, and the bulk flow movements, there's those three theories that we've just discussed. And you can very clearly see here the water potential gradient from a less negative to a more negative, from a high water concentration to a low water concentration. So let's get into the water transport in plants. So SPAC. I don't know how. This is such a good question, such a good essay question. I don't know how to make it more obvious. You need to understand SPAC. You need to understand what processes are going on. Please, I cannot stress SPAC enough to you. Okay, so we're starting off with understanding that it means soil plant atmospheric continuum, and diffusion or osmosis, and bulk flow. Now remember, let's stay with osmosis for a moment. We're just getting some terminology out the way. Remember, we discussed in the beginning video, we discussed about the fact that water can move via two ways within the cell: symplastic or apoplastic. Symplastic inside the cell membranes and apoplastic outside the xylem, in the cell walls or towards the xylem. It's a nice picture. And these aquaporins over here, this beautiful plastismata, this aquaporins over here, they are an integral membrane protein that forms the selective or water selective channels across the membranes. So that's really going to be what allows osmosis to take place from one cell to other. These aquaporins, let's zoom in on an aquaporin, because we understand that water can flow, some plastic or apoplastic. We covered that in the first section. So let's zoom in on an aquaporin. Aquaporin is a protein. Remember, your cell wall's made up of these membrane bilayer. It's really those lipid kind of things. Remember, we've got a polymer, lignin polymer complex that's really making the cell wall nice and strong. And yes, it can flow through the cell wall, but not as effectively as through the aquaporin. So you'll take more energy to get through the cell wall than you would through the aquaporin. So here's a beautiful picture of a cell, that nice vacuole over there, beautiful chloroplasts over there, and you've got the beautiful cell wall, and you've got aquaporins. So you'll have aquaporins over here, you'll have aquaporins over there, you'll have aquaporins over there. You have plasmodesmata is the region where the aquaporins are. So there's an aquaporin there, and this region is called the plasmodesmata. You'll have plasmodesmata over there, up at the top. Anything that's connecting cells around it, you'll have a zone of plasmodesmata. We zoom in on it, and we have this beautiful protein, this aquaporin, and you can see it allows water flowing a lot easier than it does through the actual cell wall. So these aquaporins play a very important role in getting the water through efficiently and effectively, not wasting energy. So let's just have a look. This is a nice picture of an aquaporin, how it would look in the cell and the water molecules moving through. And you can see in these water molecules, you've got a bit of hydrogen bonding there. So because there's a bit of hydrogen bonding, it's moving nicely through there, through this aquaporin. So what I expect you to take away from these 3 slides: SPAC, soil, plant, atmospheric continuum; 2 ways water flows: osmosis, bulk flow. Diffusion is correct, but osmosis is more correct. The water can flow, can move via osmosis through symplastic or apoplastic pathways. And aquaporins are an essential membrane protein that will really help the movement of this water more effective and efficiently. So then we get onto this cohesion-tension theory. Remember, I explained to you that this is one of the theories that water moves. Remember, positive root pressure, cohesion-tension theory, and transpirational pull theory. Now, when we say cohesion-tension theory, you guys can really do this at home. It's very easy. Okay, I'll tell you guys a story, but you're not allowed to laugh at me. Well, if you do, I'm not going to hear you, so it's fine. My first year of PhD, you tell if a seed is alive by whether it sinks or not. If a seed sinks, it's generally considered alive. If it floats on the top, we generally consider it dead. Just a quick way to test if a plant is alive or dead, or seed is alive or dead. And here I was, I'm working with Amaranthus dubius. Write it down, have a look at it. It's an African leafy vegetable. It's known as maroch, and it's just wild spinach. And go have a look, Google how tiny these seeds are. So here's Candyce, first year of PhD, very optimistic, throwing these seeds in the water, and they're not sinking. And they don't want to sink. And I'm thinking, what am I going to do? So I go out, I collect another batch, I come back, same story. And I think, no, something's wrong here. So I go back and I collect another batch, and I come back and I do it again, and they're not sinking. And eventually, after 2 weeks of struggling with this, I go to my supervisor and I say, help me. Like, what am I going to do? I can't find living seeds. And I remember my supervisor looking at me and going, really? Really? And I was like, what are you talking about? And he took a bit of sunlight liquid, popped it on his finger, touched the water, and the water molecules obviously broke, and there went all my seeds sinking to the bottom. And it was really a moment of hang your head in shame, Candice, because how? But yes, I was a brutal, cruel trick was played on me by this cohesion-tension theory, and that is why I will never forget it. So, as you can see here, the paper clip, when you put it down gently, it rests on top of the water. That's what was happening with my seed. The seed was not strong enough, or it wasn't heavy enough to break the hydrogen bonds that have created a film on the top of the liquid. So that's how you get these little insects floating on the water. They're so light that they don't break the surface tension that water creates. And water creates the surface tension because of the properties of water. The properties of water really allow it to create the surface tension. So the surface tension, the tension of a surface film caused by the attraction of the particles. So remember, the positive and the negatives coming together and forming those hydrogen bonds, it creates a film. And then you've got cohesive forces, which are between the water molecules, and adhesive forces between the water molecules and something else, another surface, other than another water molecule. So cohesive, same, adhesive, different. And generally, really, all I had to do was add sunlight liquid. So you can do this at home. Go grab a bowl of water, paper clip, a pin, anything, and try and get it to float. And then just take a bit of sunlight and put it on the water close to it. Don't touch it, just close to it. And you will see the sunlight comes in and it breaks those weak hydrogen bonds, which will cause the pin or the paper clip to sink. Alternatively, you can come and just whack it, and then the force that you create will break the hydrogen bonds around it and allow it to sink. Okay, so it's the properties of water that allow it to stick to itself, cohesive, because the opposites attract and because they create that hydrogen bond. We really get a strong thin layer that if you are small enough, if your surface area is small enough and light enough, you could essentially float on water. So if you have a look here, this is just a diagram of what I'm trying to say. So you've got a water molecule, and you know that the positive and the negatives attract and they form these weak hydrogen bonds. So you have a look here, you've got a nice balanced forces here. It's attracted to this water molecule, it's attracted to this one, and this one. It's attracted to there. It's starting to attract here, and then we've got unbalanced forces at the top. And because we have these unbalanced forces, we create a film up at the top because they're all stacked nicely strong together. So this is why, by the way, if any of you have watched the NASA, this is why when you have a look there, the water molecules or the water appears round. It's because of these positive these forces pulling everything and trying to get equilibrium that it forms a circle. So if left in space, these water molecules would come and they would come around and they would tut, and then would form that nice circular shape that you get in outer space. You can also see this on a, they say penny, I would say 50 cents. Put a 50 cents or a 5 rand if you have 5 rands laying around, and just keep dropping water droplets on them until you'll see this nice circle. It's adhering to the penny or to the 5 rand, and you'll see this beautiful round circular shape coming on it too. This is also why, have you ever wondered why raindrops are the shape that they are? So think about it. A raindrop really looks like this, or a tear, whichever. If you're feeling a bit down today and you want to cry, that's fine. um, Tear or raindrop, you've really got a circle shape down at the bottom and then this point up at the top. And this is because the water molecules are coming together and they're attracting to one another. And because they're attracting to one another and they've got nothing else to attract to, they just attract to each other, they form a circular shape. And obviously the force of falling down, gravity, will create that point up at the top. So when it has the ability to adhere to something else, such as a penny or a pool or a bowl or a beaker, it will rather create a dome shaped on the top. And this is why things float, because of the unique properties of water. So let me just check the time. Okay, 55 minutes. Great. So I think I'm going to stop it there, and then I will do the next slide. Actually, yeah, I'll stop it here. We'll discuss capillary action in the next lecture slides, on the next set of videos. So I hope everything is nice and clear. It's the properties of water that give us the ability to understand this cohesion-tension theory. Okay, stay safe. I will see or hear you guys or talk to you guys in the next video.